All right. So in this particular discussion, we'll look at the parts of the renal tubules. That is the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. So these are the structures shown here. This is a proximal convoluted tubule. That is a loop of Henle. And this is a distal convoluted tubule. Needless to say, the two convoluted tubules, proximal and distal, are both located in the cortex together with the renal corpuscle. That is the renal corpuscle there. But the loop of Henle, it's partly in the medulla and partly in the cortex. So the first part of the loop of Henle, we call them the straight tubules. This one here, straight. So these are straight tubules. These are the first part of the loop of Henle. Then the loop of Henle descends into the medulla. You can see the zone here dividing the cortex superior and medulla inferior. That is the medulla, that is the cortex. So the loop of Henle curves into the medulla and then back into the cortex. Okay, so we'll start with the proximal convoluted tubule, which is the first part there. We did a discussion on the renal corpuscle, and I'll refer you to that discussion. So I'll use a diagram to explain this part quickly. So first of all, we're looking at the type of cells found in the proximal convoluted tubule. So in the proximal convoluted tubule, we have cuboidal cells. Equally, in the distal convoluted tubule, we have cuboidal cells. Now, the difference is amount of microvilli. Okay, so we have more microvilli in the proximal convoluted tubule than in the distal convoluted tubule. If you can check, those are microvilli, and we call them as brush border. So where there's extensive microvilli, that structure is called brush border. So we have short and sparse or few brush border in the distal convoluted tubule than in the proximal convoluted tubule. So in terms of absorption, we expect extensive absorption to take place in the proximal tubule than in the distal tubule because of this specialization we are seeing on the cuboidal cell. Remember that cuboidal epithelium is specialized mainly for absorption, but can also perform some functions of excretion or what we call secretion. All right. Now, in addition to that, we see that the cells of the proximal convoluted tubule are slightly taller than those of the distal convoluted tubule. So we call this as tall cuboidal epithelium. Then this is cuboidal epithelium, or simply short cuboidal epithelium. Okay, tall cuboidal in the proximal, short cuboidal in the distal convoluted tubules. But the two parts of the renal tubules perform a similar function, that is proximal and distal. All of them are involved in the process known as selective reabsorption. That is a second stage or process through which urine is produced. The first process we said is glomerular filtration, which is influenced by the pressure found within the glomerulus, then followed now by the process of selective reabsorption, where nutrients and other molecules found in blood are being exchanged by the fluid found in the renal tubule. Okay, so explain that in a short while. All right, so that is a proximal convoluted tubule. This is distal, more or less the same epithelium. It is simple cuboidal epithelium with microvilli. Now we observe that the loop of Henle, which is shown here, that is the loop of Henle. We say the loop of Henle will have two limbs. This is a U part, this U part here. We see that it has a descending limb and ascending limb. This is descending, is going into the medulla. That is ascending, going back into the cortex. So two limbs, ascending limb and descending limb. At the same time, the two limbs are further divided into two parts. So in total, we're going to have 4 parts of the loop of Henle. So the descending limb has thick limb and thin limb. You see that this is thin then this is thick. Same with the ascending limb. That is thin, that is thick. So the difference here is that the proximal convoluted tube begin as simple cuboid epithelium all the way up to the thick descending limb. Then there's an abrupt change of epithelium from simple cuboid epithelium to simple squamous epithelium. So this part, the thin limb, is lined by simple squamous epithelium. Okay. So thin descending, thin ascending, both are lined by simple squamous epithelium. Then again, we see another abrupt transition from simple squamous epithelium in the ascending limb to simple cuboidal epithelium of the ascending thick limb. So there are two changes or transition points here. There and here. This part is simple cuboidal. That part is simple cuboidal. Then this whole thin part is simple squamous epithelium. Okay, and you and I know that wherever there's simple squamous epithelium, we expect some passive movement. You don't need a lot of energy there. But where there is simple cuboid epithelium, that part, this part, the distal convoluted tubule, proximal convoluted tubule, all those areas will require some form of active transportation. So it's not the entire renal tubule that undergoes active transportation. The thin part of the loop of Henle undergoes or transports molecules under passive kind of transportation, which can be diffusion or simply osmosis. But here, in the thick ascending, the thick descending will require some form of energy to transport molecules across. All right. So throughout the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, we expect selective reabsorption to occur. All right. So that is loop of Henle, thick descending limb, thick ascending limb, proximal convoluted tubule, distal convoluted tubule. Okay, then the last part is now the connecting tubule. This is a connecting tubule shown here. Connecting tubule, which is later connected to the collecting duct. So each nephron is attached to the collecting duct by a connecting tubule there, which is also lined by similar epithelium, simple cuboid epithelium with some microvilli. So that's all about the loop of Henle, the proximal and distal convoluted tubule. So I want to show you something. So this is selective reabsorption now. So what is happening in selective reabsorption? First of all, we need to bring that structure here. This one, I want you to have this in mind, that the entire proximal convoluted tubule, distal convoluted tubule are surrounded by capillary. This part here, the distal convoluted tubule, is surrounded by capillary. This part, the proximal convoluted tubule, are surrounded by capillary. Equally, the loop of Henle are surrounded by capillary. So when we're talking about selective reabsorption, which is a second stage in urine formation, we are talking about exchange between these capillaries, those in red, those capillaries, with what is passing through this renal tubule. This is renal tubule. From here all the way to there is a renal tubule, then connected to collecting duct. So glomerular filtrate is formed in the Bowman's capsule, then flows through the renal tubule. So the exchange is between plasma, which is found inside these capillaries, and glomerular filtrate, which is passing through these tubules. So we expect that exchange. Now let's go to the previous diagram that I showed you earlier. This one here. So have in mind that even though here we are seeing that this is the outside of the renal tubule, this is a renal tubule inside here, then outside is the parenchyma of the kidney or the kidney interstitial space. Then around this, there are capillaries. Okay. So there will be exchange. Molecules in blood, because there are blood vessels here, will shift from blood vessels into the kidney interstitial space, then those molecules will be absorbed here into the renal tubule. Then at the same time, various types of substances will be moving or diffusing or being transported from the renal tubule into the kidney interstitial space to enter into the blood vessel. So the main thing to remember here is in the blood vessels, which are next to this renal tubule, what is being transported out of them is waste matter or waste substances that were not earlier filtered here in the Bowman's capsule. So they will move from the blood vessels into the tissue of the kidney and then finally enter the renal tube, because this is where waste is supposed to be found so that urine can be expelled as waste matter. At the same time, useful elements that are here that were filtered during filtration, the body still needs them and we don't want to lose it in form of urine. So all the useful elements like glucose, some minor proteins, water itself, which is essential to body function, will be transported out of the renal tubule into the tissue of the kidney and then finally enter the blood vessels. So throughout this is a proximal convoluted tubule. Then this is loop of Henle, distal convoluted tube. You can see that there's exchange of those molecules. So in the thin limb, we have passive transportation, but in the thick limbs, thick ascending, thick descending, we have active transportation. These two processes are essential to the final product, which is urine. So we find that urine is waste. Most of the substances that are there, we don't need them. Hydrogen ions, excess sodium, excess water, inactivated hormones, inactivated drugs, all those are moving from the blood vessels into the tissue, into the renal tubule. Glucose and all the useful elements are moving out of this into the tissue, into the blood vessels. So that process, you see that it is selective. It's not all elements from blood going there, and it's not all elements from the renal tubule going there. It is only those selected elements. That's why it's called selective reabsorption. Okay. Now we are saying the last part of urine formation is secretion. You can also call it selective secretion anyway. So what happens is that there is now removal of waste that is here. Remember, waste matter must come from blood vessel into the tissue, then enter there. Now, it's not all waste that will be absorbed quickly into there. So some of the waste matter may remain into the interstitial space and they have not yet crossed to enter the renal tubule. What should the body do to that? That is now when we need process known as secretion, the last stage in urine formation. So urine formation has only 3 stages: filtration, selective reabsorption, and secretion. So what happens during secretion is all the waste that has accumulated in the kidney interstitial spaces has to be secreted. And that waste is transported into the collecting duct here. So waste in the cortex, waste in the medulla will be secreted one way. So there's no exchange this time. It's waste moving out of the kidney interstitial space direct into the collecting duct there. So this is one-way transport system. And that stage now, urine is almost formed. So from there, urine, the product will follow through the collecting duct and reach the minor calyces, be transported to the major calyces, and finally reach the kidney pelvis and enter the ureter here as urine. Okay. So the lining of the collecting duct is the same. It is still simple cuboidal epithelium. But this time we see that this epithelium has actually. Some books may argue to say there is minor microvilli, but there's a general agreement that these cells here do not have microvilli. Okay, this is a collecting duct. Cuboidal cell, cuboidal cell, cuboidal cell. We don't see any microvilli there. And that's how you distinguish a collecting duct from the proximal convoluted tubule and from the distal convoluted tubule. Let me show you a histological diagram of the distal and proximal convoluted tubule. Then you can judge for yourself. Okay. For example, this one. Okay, so that is a collecting duct. Okay. Collecting duct, collecting duct. This is a thin loop of Henle, or the thin limb of the loop of Henle. You can see that there are flat cells there. So that is simple squamous epithelium. Simple squamous epithelium. Okay, then this is the ascending thick limb of loop of Henle, the thicker one, but it is ascending because we have a descending one and an ascending one. So we are getting this from the medulla. So this sample is from the medulla. Wherever you see those flat cells, think of the thin. Whether it's thin ascending or thin descending doesn't really matter because it is still simple squamous. But the thicker one, whether it is ascending or descending, you see that the cells are cuboidal here. Similar with collecting duct, except that here the lumen is quite smaller, and you see those fuzziness. Those are microvilli. But this one is devoid of microvilli, and therefore the lumen is wide and quite extensive than those other parts. Okay, again, collecting duct, ascending thick limb, collecting duct, clear lumen, like that. So in terms of absorption, you can guess to say in the collecting duct, absorption is quite slower as compared to the proximal and distal convoluted tubule. In addition to that, not just having microvilli, we call it convoluted because it's highly tortuous. It makes several turns and twists. Now, that makes it a longer tube than if it is a straight tube. You and I know that the fastest route to any destination is to take a straight line. The moment you start making twists and turns, it will take a lot of time for you to reach the destination. Same thing here. These two parts, the proximal and the distal, are made to be convoluted to allow enough time for selective reabsorption to occur so that all the useful material that were filtered can be transported back into the blood vessels. At the same time, all the non-useful material that were not filtered can be now transported from the blood into the renal tubules. That's the whole essence. Otherwise, if the tubes were straight, like it is shown here, you find that it would have been shorter and effective urine formation would have been a challenge. You find that excretion of waste was going to be slow, and therefore waste would accumulate in blood and cause various problems to the normal physiological function of the human body. All right, so that's all about the renal tubule. Okay. Away from that, I would like us to explain or to discuss what we call macula densa. I may have hinted on this in the other discussion, but here we'll look at it a bit more detailed. So we saw that the loop of Henle descends into the medulla and then ascends back into the cortex. Goes into the medulla, back into the cortex. Now, as it goes back into the cortex, you see that the distance between the proximal and the Bowman's capsule becomes closer. We call this distal because it is far away from the renal corpuscle. But there's this part here where the distal convoluted tubule comes closer to the Bowman's capsule. You can see that part, this part. We call this part as macula densa. It is a specialized area of the renal tubule in that the cells found there, that is the cell. So we find specialized cells in the macula densa. This is renal corpuscle, Bowman's capsule around, and this is a glomerulus. Then that is the distal convoluted tubule. Now the cells at that point in blue, where the distal convoluted tubule comes close to the glomerulus or the Bowman's capsule, these cells are specialized cells and they are called macula densa, or simply dense body, if you like. Okay. So what is the importance of this, dense body? Number one is that the cells found there are columnar cells. Okay, we established that the distal and proximal convoluted tubule are lined by simple cuboid epithelium. But here we see that that part called the macula densa, the cells there are columnar cells. Okay, and therefore now form a structure known as juxtaglomerular apparatus. Juxta means closer. Glomerular is the glomerulus, which is inside there. So this part is closer to the glomerulus. Even though it is part of the distal convoluted tubule, it comes close to the glomerulus, hence the name juxtaglomerular apparatus. What is it that is so special, apart from just having columnar cells? Okay, number one is that these cells store and secrete a hormone known as renin. So those cells on the macula densa secrete renin. Now, these cells, the juxtaglomerular cells, excuse me first. Yeah, macula densa, like we are saying, is columnar cells. Yeah, these cells are specialized to produce and secrete renin. So renin is secreted in response to the volume of blood and the osmolarity of blood. Volume, we're looking at the amount. Then osmolarity, we're looking at the amount of dissolved element in blood. Okay, so blood may become dilute, blood may become concentrated, depending on the fluid intake or any other, hemodynamic changes in the human body. So depending on the osmolarity of blood, how, for lack of a better term, how thick or how thin the blood is, renin will be secreted. So renin is an important substance that is essential in controlling blood pressure and fluid volume in the body. So renin activates angiotensinogen, which is produced by the simple squamous cells found in blood vessels. So when this hormone is produced by the juxtaglomerular cells, what follows next is activation of angiotensinogen to angiotensin 1. Then angiotensin 1 is further converted to angiotensin 2 by an enzyme found in the lungs. The blood vessels in the lungs secrete an enzyme known as angiotensin converting enzyme. That enzyme converts angiotensin 1 to angiotensin 2. It is angiotensin 2 that leads to production of aldosterone. Aldosterone is produced by the adrenal gland. These are small glands located on the superior part of each kidney. Aldosterone leads to sodium conservation. So the body will store sodium, at the same time will store water. Then angiotensin 2 causes vasoconstriction so that more blood can go to the glomerulus. Okay. So that's how it's able to control, blood pressure in the body. So that's how the macula densa is important, because this acts as a sensor testing the osmolarity and the volume of blood passing through the kidneys. Okay, so we call that as a juxtaglomerular apparatus. Juxtaglomerular apparatus. Macula densa are able to pick up osmolarity of blood. Then the juxtaglomerular cells secrete renin. Okay, and then we see that there is conservation of water and vasoconstriction. Okay, finally, I can now look at the functions of the kidney. Mainly is urine formation, and some of them we tackled them in the introduction to urinary system. So mainly this is what I needed to talk about. Okay, next time we'll look at the ureters and then also talk about the kidney and the urethra. For now, we can leave it here. Unless there are questions, you can still get in touch with me and we'll be able to discuss. Thank you for your attention.